We would like to warmly welcome you to our today's webinar, Solving Ongoing Difficulties in Membrane Protein Purification Using Established and Brand New Copolymers. My name is Lena. I'm a project manager here at Cube Biotech and responsible for our service projects. Today, I will guide you through this webinar. Here at Cube Biotech, we're excited to work with membrane proteins each and every day. And over the years, we have accumulated extensive knowledge and experience about membrane proteins. And synthetic copolymers have become a milestone in this process. And today, we would like to share what we have learned so far with you. Our speaker today is Dr. Philipp Hanisch. You can already see him. And he has studied biology between 2010 and 2019 at the University of Düsseldorf. He did his PhD in the lab of Hans Hegemann, functional genome research of microorganisms, where he investigated the chlamydial membrane proteins LIP and PMPs that are involved in the infection pathway. Philipp has gained a profound knowledge about soluble and membrane proteins, and he has many years of experience with the expression and purification also of tough targets. And in this context, he is highly experienced in working with detergents and MSP-based nanodiscs, but also became a great fan using synthetic copolymers in the last years. And you will definitely see that during his talk. If you have any questions during Philipp's talk, he already mentioned it, please feel free to use the Q&A function in the menu board, since your cameras and microphones are automatically deactivated. We will try to answer as many questions as possible after the talk, but of course we'll also answer all remaining questions by email. So now I would like to wish you to enjoy the webinar. And yeah, Philipp, it's your turn.
Yeah, let's go. Yes, perfect. Okay. Hi, everyone. I hope you can see me well. I will just share my screen and then we can already get started. Okay. So if we're lucky, you can now see the starting screen. Lena, can you give me a thumbs up? Is this working? Perfect. Okay. Then I'm ready in a bit. And then we can start over. Okay, yeah, hi, I'm Philipp. Lena, thank you very much for the super nice introduction. Indeed, I'm hyped to be here and to talk about copolymers and of course also MSP and detergents. And I hope you're just as excited as me and just start over. Okay, what you see here is basically the roadmap we're going to travel along during the talk. So we're going to talk about several problems you're going to have to face on the way from the tech choice to successful membrane protein purification. And we're going to give you tips and tricks and explain you what we experienced over the last years in working with these indeed have very tough targets. But before this, I'm going to start with the introduction. And this starts with the human cell we can see here. So in the human cell, we obviously either have soluble proteins, which are 76% of all encoded proteins, so the majority, and all different membrane proteins, which are around about 24% of all proteins. This can be either monotopic proteins, bitopic membrane proteins, or multi-span membrane proteins, either in the form of alpha helical or beta sheet forming proteins, or we can have lipid anchored proteins. In total, in humans are 6,718 proteins encoded, but they are not expressed all the time. We have a basal expression of around about 4,000 different membrane proteins, and then depending on environmental factors, this number can differ. Okay, what you see here is a graph of the number of unique structures of membrane proteins over the years from all species. In 1985, the first membrane protein structure was solved via X-ray crystallography using detergents, which were invented in the 1980s. And you can see here, over the years, we can see that the numbers of unique structures definitely do increase, but still there are not many structures which are solved. In the year 2020, we had around about 130 different structures. What you can also see, and what I found very fascinating, is that even though X-ray crystallography still plays a major role, EM, especially cryo-EM, is slowly taking over and is taking a big bunch of all the unique solved structures. But nevertheless, only 3% of all structures in the protein database so far are membrane proteins. And this is of all species. So you can imagine that there is still much room for improvement, and the majority of targets has not been solved so far. Okay, we at Cube, why we are interested in membrane proteins, even though they only make 24% and only 3% over all species are so far characterized. This is because the majorities, 60% of all human drug targets are indeed membrane proteins. This can be the famous GPCRs, ion channels, we have membrane-associated proteins, solid carriers, or receptors. And these can be for pain or several different sicknesses in humans. So they are very important to know exactly what the structure is, so we can find ligands which work the best for this. And working with membrane proteins is obviously not easy. Everyone who ever worked with membrane proteins could experience this firsthand, because they are embedded in the lipid bilayer, and today we know that this lipid environment is crucial. It's super important that we try to keep the natural environment intact and change as little as possible. Starting already at the expression level, it's quite hard to overexpress membrane proteins to get them in sufficient levels to then extract a good amount of membrane protein. We have to be careful that we keep the native folding intact, and also to keep the lipid environment intact. And last but not least, we also have to take care of the stabilization process. So when we solubilize the protein, we also have to keep it in a stable state that is not disintegrating, especially if we are talking about big membrane protein complexes. The take-home message for you definitely is that membrane proteins are extensively interacting with the lipids. It's a synergetic relationship which stabilizes the protein and often modulates its function. So it's really, really crucial to keep this membrane environment intact. Okay, before we start with full blast into the webinar, I want to tell you a little story about how it all started here at Cube. So since a few years, we are really interested in the synthetic copolymers, which have been introduced in 2009 the first time. And we had several different candidates. I call them here GPCR candidate A and candidate B. And I just want to tell you how we started trying to work with these synthetic copolymers. So we made a solubilization screening with, back in the days, several SMALP and several dipmers. And we could see that you don't get the same solubilization efficiency with every polymer. In this case here, SMALP 200 was the best. So we decided to go on working with the SMALP 200 to solubilize and stabilize our GPCR candidate A. We then made a big pH screening, and we could see here that under the condition of pH 8.0, we got the best efficiency. So we went on working with this. We also could see that this is our total solubilization, and this is what we could recover from the soluble fraction, that we can really recover enough material to go on working with. And then, unfortunately, we saw that we have a big part which is lost in the flow through during the purification process. So unfortunately, in the end, in the elution fraction, there was very, very little left, which was not detectable in Comassi. And we failed at this point. We were not able to elute a proper amount of this candidate A GPCR. And we tried also with a different candidate. And what we could see here again is over the course of the purification, we lost and lost and lost more material, that in the end, again, nothing was left. And we couldn't recover enough protein also here. So this was kind of sad, and we were not super happy about it, as I guess you can imagine. But now, a few years later, we stand here today, and we can proudly tell you that now we are indeed able to recover actually enough protein. And this is here P2X4. It's an ATP-gated cation channel. It's also a multi-span membrane protein. And we were able, with AASTY and ultrasolute in this case, to really recover enough protein to even go into EM and into cryo-EM. And here you can see examples of a normal negative stain electron microscopy. And we could even fit what you could see there in the structure, which was already solved. So right now we are trying to resolve the first cryo-EM structure of P2X4 solubilized and stabilized in new synthetic copolymers, which is super exciting. And I'm very happy to stand here today to tell you this and to be part of all this. Okay, so now that you know that also we are not perfect and we can also fail and we learn, let's start with the first part, which is the tech choice and the expression, because we saw something very interesting, which I didn't know before myself. So it doesn't matter if you work with prokaryotic cells or with eukaryotic cells. Obviously, if you have a human membrane protein, I can just kindly advise you to use human cell culture. And if you want to overexpress this, it's really important you make a huge mind about the tag position and which tag you want to use. You know, you can use a his tag, a GST tag, you can use antibody tags like flag, or especially the row tag we are using here all the time, and I'm very happy with. And we could see that there is definitely an effect of expression and detection level, which I would like to show you now. Okay, here we have an example, again, a GPCR, which is N-terminally tagged with a flag tag and C-terminally tagged with a row tag. We have here Western blots after 24, 48, and 72 hours of expression. And as you see, you don't see anything, which means we couldn't actually express and or detect our protein of interest. Interestingly, if you now just switch the flag tag to the C-terminus and keep the row tag at the C-terminus, now you get a nice signal for the flag antibody, and you get an amazing signal after already 24 hours, but even better after 72 hours for the row tag. So this was quite stunning to me, and I did not expect that just changing a tag can completely change your expression and or detection level. And I have another example for you, again, a GPCR with a 6 times his tag in the N and a row tag in the C. We can see this is 10-minute detection for row. This is really not proper. And the 3-minute detection for his, in our case, this is also not proper. And I would not like to use these pellets to go then on in the purification. Because as you know, if you start with little, you will end up with none. And if you start with a lot, you will end up with a bit. And a bit is what we're looking for. So if you also switch this here, the his tag to the C-terminus, we get an immensely better increased signal. 25 seconds for row after 48 hours is amazing. This is what we want to see here. And also with the his tag, we could really nicely detect this. Interestingly, you get rid of the row tag, the his signal also decreases dramatically. So what I want to give you as a take-home message is take time, think about the tag, try some stuff out, because you will regret if afterwards you find out a different tag would have given you a better expression level or a better detection level. And this brings us to the first take-home message, which is tag choice and position influence expression and detection level of recombinant membrane proteins. So it's worth to spend the time and to go for this. Okay, let's move on to the solubilization, one of the most critical parts in membrane protein purification. So there are 3 options to solubilize a membrane protein. As I said, membrane proteins are obviously located in a membrane, so it has a big hydrophobic core, which we have to protect against aqueous solution around. And the first thing which scientists came up with in the 1980s was detergents. They worked great, and they are still used today. And I will show you now the advantages and disadvantages of detergents. Okay, what we can see here on the top, we have native lipid surrounding and we have a non-native lipid environment somewhere over here. Obviously, the membrane protein really loves to be in its native state, embedded in the original membrane. And if we use detergent, we usually do strip away a majority of these native lipids. Advantages with detergents are definitely some detergents are able to solubilize even proteins with high transmembrane counts. We have a high solubilization efficiency and we have quite a fast solubilization. Sometimes solubilization times of 30 minutes up to 1 hour are already enough to solubilize huge membrane proteins. A big disadvantage, on the other hand, is we fall short on mimicking the lipid bilayer. And here I'm talking about molecular composition, the lateral pressure, and also the thickness of the membrane, which can be a crucial factor in folding and in function of membrane proteins. Which is kind of an annoying point for me, is every buffer always needs to be supplied with detergent concentration above the CMC. If you forget about this just once, it can ruin your whole protein purification. So you have to pay attention that you always supply your buffers. You have to create separate buffers with different concentrations during the purification process. And yeah, it's just quite much work. And one of the most nasty parts here is definitely that detergents do interfere with a bunch of diagnostic assays, for example, ELISA, and you cannot use your protein in detergent for this. You have to get rid of the detergent before. Okay, people knew about this. So in 2002, they came up with the multi-scaffold membrane proteins. This is a protein ring which can, together with lipids, form a nanodisc around your membrane protein of choice. And it definitely is already better than detergent for the lipid environment, because we actually do have many different lipids here. You obviously have all the advantages of detergents and solubilization, because MSP nanodiscs cannot solubilize. You have to use your detergent of choice, where there is a broad selection for every liking, and then you have to introduce the MSP nanodisc with your lipids and get rid of the detergent so the nanodisc can stably form around the hydrophobic core of your membrane protein of choice. You can freely pick which lipids you want to use, so this is just up to you, and there are different sizes with or without a tag available, which you can also purchase from our webpage, by the way. A disadvantage is that the lipid environment is most likely not native, because often we just do not know what lipids are around our membrane protein of choice. If we just talk about microdomains in different membranes. You have additional processing steps, which is time consuming, and you're gonna lose much of your protein which you had in detergent before. So this is really a step you have to know. You will lose protein if you do that, and you cannot always be successful because you have large hydrophobic regions in big membrane proteins, and this can be problematic since the MSP size is limited. I know there are already different solutions for that with DNA rings and bigger nanodiscs, but still it is crucial which size you have to use for this. And in 2009 then, the first copolymer, the SMALPs, they were introduced, which now brings us to this advantages and disadvantages comparison. The copolymers are as close as you can anyhow get today to the native lipid environment, because they directly cut out your membrane protein of the lipid bilayer and stabilize it in its natural surrounding, which means the native lipid environment is preserved, and we can also solubilize functional complexes as units. This has already been shown in several publications. You don't need to modify your buffer anyhow. You just use one buffer for the whole purification and for the storage. It's very convenient. You have a one-step solubilization and stabilization. This is also unique with MSP nanodiscs. You first have to solubilize, then you can stabilize. But here, everything is done for you by the copolymer, basically, in one step. It's pretty easy and also fast to learn of a workflow, so also suitable for students which are not super experienced in the work. and copolymers do not interfere with diagnostic assays. So here we go, diagnostic assays is definitely doable. Disadvantages, of course, there are also disadvantages, are that you often have a lower solubilization efficiency compared to the big range of detergents. Detergents are still powerful and are still used up to this day. So this is something you have to deal with. And also often you have a slower solubilization. We are trying already to reduce that. We tried successfully already to do 3 hours, and we are currently trying to even get lower, but it will not be as fast as with detergents. Okay, today at QBiotech we have 4 different polymer classes in our storage. This is the original 2009 introduced SMA or SMALP nanodiscs. Shortly after the dip masks were introduced, we are also selling different kinds of dip masks. And just this year, we introduced 2 new classes. These are the AASTY and the ultra-solute amphipol. And they all differ a bit in structure and in behavior. And I'm going to explain you in detail what the advantages, disadvantages, and the differences are. But first, how are copolymers actually working? So you can use them either in a cleaned membrane, or you use just a normal cell lysate, for example, a 9000G supernatant. Or what's super interesting is you can just use these copolymers on whole cells. So yes, you heard it right, they actually can solubilize membrane proteins and stabilize them directly out of whole cells. So not even a lysis step has to be performed. And you cannot even do this at 4 degrees. You can also go higher in temperature. You just add them to your membranes or to your cells, and they will cut out a bit like a stamp. We always talk about that, and stabilize all membrane proteins and solubilize all membrane proteins which are found in the membranous environment. And then via your affinity chromatography with a tag you introduced in your overexpressed membrane protein of choice, you will then be able to select for your protein of desire. And now let me tell you how those copolymers are actually built and what X and Y means. So here in the example of SMA, but this is true for all copolymers, we always have an X part and we always have a Y part. And the X part is the hydrophobic part, which is important for the solubilization of the membrane protein. The Y part is the hydrophilic part, which is important for the stabilization of the membrane protein. In general, you can say that smaller polymers are more aggressive and better in solubilizing, while bigger polymers have a higher membrane protein stabilization. The X part, here it's important to tell you that the flexibility influences the solubilization, and also the density and nature of these hydrophobic groups can influence the solubilization efficiency. You're going to see that later, that different polymer classes have sometimes different X parts. Also, here at the Y part, the density of charges can influence the stabilization efficiency, and a crucial part is also that copolymers are only suitable in a certain pH range, which is above 6.5. What you can see here, if you go lower as pH 6.5, all copolymers here are 3 different AASTYs and a SMART 100 shown. They start to protonate, and this means they will not be soluble anymore, and they will fall out of your solution as a white powder. This is something you don't want. If we have the white powder there in your solution, the copolymer won't be able to solubilize anymore. So please always stay above a pH of 6.5. Let's start with SMA. SMA stands for styrene. This is the styrene. Maleic anhydride. This is the maleic anhydride. You see it has 2 negative charges here, and it has this aromatic ring on this part. Advantages are SMA has high solubilization rates of membrane proteins. It was the first class which was introduced and is the utmost used polymer on the market. We even have a SMALP community where several working groups are organized. not only talk about SMALPs, but all the other polymers. SMALP was the first. This is why the community is called like that. We have a high diversity in styrene to maleic anhydride ratio. We sell several SMALPs, and you can always see on the number 3 to 1 means ratio 3 to ratio 1 x to y up to 1.4 to 1. And they are negatively charged, as you can see here, and small enough for excellent solubilization. They are 5 to 10 kilodalton. Unfortunately, SMALPs, because of the aromatic ring here, they do absorb at 280 nanometer, which means if you, after your purification was successful, want to determine your protein concentration, you cannot use UV vis, and you have to use different techniques for that. What many copolymers have in common is that they are prone to aggregation in high cationic environment. For SMAL, this is above 5 millimolar, except the small 140E. This is modified in the Y part and is super stable also in high cationic environments. But also the trade-off is then it's not as good in stabilizing the membrane proteins. Of course, cationic environment is not always crucial, but if you work, for example, with GPCRs, you have to add calcium. And if you add too much calcium, you're not going to be able to use the majority of the SMALPs. So the DIPMA stands for diisobutylene, here maleic anhydride, and it differs from the SMAL in the X region. The Y region is the same. Also here, please only use it above pH 6.5. We have two different molecular weights available, 10 and 12 kilodalton, and additionally, we have two less charge modifications available, which are the DIPMA glue and the DIPMA Gli. They are also negatively charged. They have a very little absorbance at 280 because they do not have the ring, and their calcium tolerance is 5 to 10 times higher compared to SMALPs. For example, DIPMA glue can withstand up to 50 millimolar calcium. Challenges are DIPMAs have generally lower solubilization rates of membrane proteins compared to other copolymer classes. This doesn't mean that DIPMA is not as good as the others. You're going to see later that sometimes DIPMA can be the champion. And DIPMA 10 and 12 are also prone to aggregation, but not in such a high amount as the SMALPs, for example, are. Okay, what you can see here is our DIPMA glue. You can see here there's a modification in the Y part, and you can see that it's compared to the normal or different DIPMA, definitely more stable and higher cationic environments. Okay, let's come to the AASTY. We can see AASTY stands for acrylic acid co-styrene, also usable above pH 6.5. And the advantages here are clearly that you can also have high solubilization rates of membrane proteins and low polymer ranges, which you can see here. In, whoops, where is my marker? In 2.5%, you get a great solubilization. This is always total, and this is what we could solubilize. But if you go as down as 0.1%, you still get a really good solubilization in this example. So it's worth definitely to give it a shot if you do your screening, or if you want to decide for which copolymer you want to go for, to also try out lower solubilization, lower amounts of copolymer for the AASTY. We have a more alternating monomer sequence in the polymer than for the 2-to-1 SMAL copolymers. This is due to the fact that RAF polymerization is used to create this copolymer. We have different ratios of acrylic acid to styrene available. These three, we have a small variant, which is 5 to 6 kilodalton, which is more aggressive, and we have a 10 to 11 kilodalton, which is a little bit more thermodynamically stable. Another advantage is that compared to other polymer classes, it is higher in homogeneity, which you can see nicely here. This is also due to the RAF polymerization. So what you get here is really a narrow distribution of size, whereas as SMALs, DIPMAs, and the ultrasolute, this is a bit of a wider distribution. This doesn't mean that AASTY works better in solubilization and stabilization, but if you are looking for something where the size is precisely defined, then AASTY is a good choice here. And last but not least, we have our newest candidate in our online shop. This is the ultra-solute amphipole. This is an acrylic acid cycloalkane. Again, acrylic acid, just like in the AASTY. And here we have a cycloalkane. Compared to the amphipoles you might maybe know, which were introduced already many years ago, here we have a replacement of the linear octyl chain for hydrophobic saturated cycles. This is what you can see here in this part. And this is the first amphipole ever which can solubilize and stabilize membrane proteins. Just like in AASTY, we also here have high solubilization rates of membrane proteins and low polymer ranges, up to 0.1%. They are also negatively charged and quite small, and we have no noteworthy absorption at 280 nanometer, which is a big advantage in my opinion. Unfortunately, so far, the behavior in cationic environment is not yet determined, but this is definitely up to come, and we're gonna support you with this information also in the near future. Okay, what we can see over the years, since 2008, 2009, that we have an exponentially growing interest in copolymer stabilized membrane proteins, which you can see here. The number of publications and patents is really rising from year to year. This is a picture from also the SMALP community. And this is especially interesting for cryo-EM, for crystallization, for drug targeting of membrane proteins, and for diagnostic assays. Now let's come to why I told you that not one polymer is always the best. We discovered that really screening is key, and every membrane protein has different polymers which work the best with this. What we see here is a GPCR, and we can see that in this example, the AASTY 1145, a SMALP 502E, and some DIPMAS worked the best. In this example, which is a 9 transmembrane domain transporter, we see a completely different picture. Here the AASTY 645 is really good, but a lot of SMALPs, a lot of AASTYs, DIPMAS, and also the ultra-solute, they work nearly similar. So it's really important that you do screen. Always remember, AASTY and ultra-solute, you can also try, if they are working well with 2.5%, maybe they are even working better at lower concentrations. This is what we also sometimes see. You lower the polymer concentration, you even get higher solubilization rates. So this is also, for you, interesting to keep in the back of your head. And if you want to try that out, and if you want to do a big screening, like I would definitely advise you, we see with every new target that screening is really important. We can also provide you with our Maxi screening kit. There you have 2 small samples of nearly all polymers we are selling. In there, they're already pre-buffered, 50 milligram per vial, and then you just add your sample, you solubilize it, and then you can directly check on blood, which is the best for you to go with. But we even went a step further. We did a huge collaboration with the University of Cologne with a mass spec department, and we created a solubilization database for you. In there, you're going to find 4,000 different membrane proteins, the one I was talking about before, which are now always expressed. And you will be able to see detergents and copolymers and how good are they in solubilizing the native expression level of these target proteins in human cell membranes. And we did this with a 2.5% copolymer or with a 1% copolymer. So you can directly see maybe a lower concentration is even better for my purpose. Please keep in mind this is native expression, so it might be completely or a bit different with an overexpressed membrane protein. But first, I would like to introduce you into this, and I picked a nice example. This is a 2020 paper where the working group solved a 2.9 angstrom electron microscopy structure from the protein SLC12A7, and they solubilized this in LMNG and CHS. And I used this because I found it quite interesting that here the AlphaFold prediction was not as accurate as AlphaFold is often stated. We can see here that it's a dimer, that it's glycosylated, and also this part was quite often the prediction. And if you now go to our website, if you click here on solubilization database, you're going to get into this window. And then if you, for example, search for the SLC12A transporter family, you're going to get all the hits of of all the SLC12A family members which are there. And then you can see that some pointed out in our experiments and some they are gray, they couldn't be found in the pellets. And you see all the different dip mass AASTY SMALPs, and you can also see different detergents and a REPA buffer. And you can directly see in the color scheme, red means very good solubilization, gray means no data, and light yellow means quite bad solubilization. You can also see already the amount of transmembrane domains. And if we now pick out the example I showed you before, we're going to get to this page. And if we zoom in a bit, you can directly get all the information you're going to need. You see 12 transmembrane domains, you see the mass, you see the subcellular location. You will get the extinction coefficient and all the important data. You see both our experiments, 2.5 and 1%, and what was the outcome. You're going to see that there's already an electron microscopy structure solved. This is the paper I just showed you, and you will already get the amino acid sequence. So this is everything in one spot, all the information the probes need. You can also download a PDF. This is up here, just click it, and then you're going to get a big PDF. And on the second page, you're going to now find all the experimental data, not only in the color scheme, but also with the numbers. 1 meaning perfect solubilization, and NaN means no data. So the closer to 1, the better the solubilization is. And if we now take a closer look, we can see that in the native expression of this very protein, LMNG works really good. So they made a wise decision to solubilize it with this. But they could have also went with amphipole 17 or 18 or SMALP 200 or 300 because they work also really, really good. And if we now compare this with a lower concentration, LMNG is still the same concentration. There was always 1% detergent used, also solubilized at the same time. And we can see that the amphipole are still working nearly as good as with 2.5, while the SMALPs are not working as good. And earlier I told you that sometimes AASTY and amphipoles are even working better. And this is also a nice example here. You see AASTY 650 was 0.42 and 2.5%, but it's 0.68 1%. So in this case, it was worth to also check the lower concentration of the AASTY to get even a better solubilization here. So very useful. Just go to our webpage, click on the button, search for your protein of desire. If you don't find it, maybe look for the whole family and check for similarities. And this can already give you a big hint also for your overexpression. Okay. And let's move on. Your second take-home message is choice of copolymer, concentration, and the temperature are crucial for each individual membrane protein. And we at Cube, we say screening is key. And I can just advise you, do your screening. This, I know it's much work, it takes time, it takes effort, but it's really worth, because you want to stand in the end there with a pure protein which is concentrated enough for your approaches, and you will not be happy or give maybe up if you do not do the screening before. Okay, let's move on to purification and dilution. Okay, what you see here is the solubilization I showed you already earlier from the 9 transmembrane domain transporter. And we see that it's quite similar. Okay, if you now go over the whole purification, you will be shocked that in the elution you see this looks completely different. Why is that? Why is DIPMA here so nicely eluted and my AASTYs, most of my SMARs, and one of the ultrasolute are not working so good? And this is something we see often. Now you would say, oh, okay, then I only just use DIPMA because they work fine. Yeah, okay, we also thought that, and we tried different membrane proteins, and we see here in the solubilization, the DIPMAs are amazing. This AASTY is really good, ultrasolute is okay. If we go to the elution, it turns completely around again. And here, a few of the AASTYs are really, really good, and the ultrasolute is amazing. The SMALP 502E, where there was nothing here, looks really promising, and compared to the rest, this is not so good anymore. So why is that? This is one of the main questions we were asking ourselves for the last 2 years. Why is it that there is such a huge gap? And this can only be, to our opinion, that either the polymer interferes with the detection on the Western blot, or it interferes during the binding process for our affinity chromatography. And here also in the illusion on the Comasi, you can see that is really true what I just showed you on the Western blot. Okay, and I want to show you an example where you can see that really you lose a lot during your purification if you have much free copolymer in there. Again, here, a 9-transmembrane protein, ER membrane protein, we can see that it was solubilized with 2.5% SMALP, 200, DIPMA 10, and AASTY 1155. This is all comparable here. It was all done with MBP. And if we now put this on our affinity column, what we're going to experience is that we lose a bunch of our protein of desire, the protein we worked so hard for in the flow through. And this is true for nearly all copolymers, for nearly all resins, and is always different depending on the target protein you're now working with. It's really always different. And then you end up with less protein. And interestingly, as I showed you before, even if solubilization efficiency is comparable, here at SMA it was even better. SMA is significantly lower than the AASTY 650 in this case. And this is really the harsh truth. So we worked really hard on this, and your third take-home message is now the copolymer type and polymer concentration can impact the binding of your target membrane protein. It's not always the same, it's always different, and this can be nasty. So you have to get rid of your free copolymer. You need a certain amount of polymer to solubilize all your membrane protein of desire. There definitely is going to be free copolymer left in the solution, and you have to get rid of this. And there are two options. Option number one is you dilute up to 100, which means if you have 40 milliliter in the beginning, you're going to end up with 4 liters. 4 liters is not easy to work with. If you have a column, if you do it with 1 milliliter per minute, you can calculate yourself. This is going to take a whole night to load everything on your column. But if you take MAC beads, this can be faster. I can advise you if you want to go this route, and if you want to be sure, and if you want to dilute 1 to 100, use MAC beads. And this is going to be very much helpful. But yes, we at Cube, we were not really happy with the solution because we always want to make it best, obviously. So we worked super hard on creating a product. And today you are the first in the world, actually. I can tell you about this. This is our brand new copolymer depletion matrix. So we called it Polyhunter, and this is like the first product I'm also involved in launching. So it is my little baby, and I'm very, very happy to talk to you today about this. It's super cool because you are able to deplete up to 99% of the free copolymer. This is an example of AASTY. It absorbs at 260 around in 2 peaks, and you can see there's nothing left. So really, if we use this depletion matrix, you're gonna get rid of nearly all your free copolymer. And now you say, and what about my formed nanodisk? And I can say, keep calm. You're gonna recover them. You're gonna recover 100% of your formed nanodisks, and they stay intact. That is super nice. And we also tried this in the purification. You can see here, this is your supernatant before Polyhunter. This is after it ran over the column, looks the same. You do your purification, you have lots of protein lost in your flow through. You have a little less left lost in this one, and you can even recover more protein if you let it run over Polyhunter than if you do the purification without. And this is now an example with an AASTY which did not change so much the purification with the free copolymer inside. But if you really have big issues with losing sometimes nearly all your formed nanodisk in the purification process, then this can really be a game changer. And I can just advise you to give it a shot. If you do not want to dilute, and if you see that you have these problems, give it a shot. Use this. You can renature it up to 5 times. So you can use it over and over. And this is really a great help for me. And I'm super, super happy to now show you that you can, from now on, actually buy this from our website. We have them as MAC beads and as agarose. And if you have big bunches, the agarose is still very suitable for this. Okay. So that in the end, if you listen carefully and if you are brave enough to try all this and if you stick to it and you bite through, you're also going to be successful in your membrane protein purification, and you're going to recover all this. You're going to get pure protein after size exclusion. You're going to make that cryo-EM structure, and you're going to be having that Nature paper or Cell or whatever you desire. Actually, do your assays, and you're going to be successful. Okay. Your last take-home message here is now that comparable solubilization efficiencies do not always correlate with the membrane protein elution quantity. If you have this, use polyhunter or dilute it. And one of the last things I want to tell you now is, of course, if you went all this way and you have your elution, you still have to be paying attention to the details. And this is what we see often, the people getting a bit lazy in the last spot. Don't be like this. What we have here is our set chromatographs of all the P2X4 alloates we created with detergent, in this case DDM-CHS, and we can see it elutes here, and that is the maximum where the black arrow is. And if we now compare this to the ultrasolute, you can see it runs significantly more in the front, which we think is due to the conserved lipids you have in there, which we now know are super important. We want to keep them there. And sometimes you also have a bit of a broader peak, for example, here in this AASTY. And if you're very unlucky, like I was here in this purification, you also have a lot of aggregates here. So please do a sec to make sure that you don't put aggregates on your grids, or you don't use aggregates in your diagnostic assays, because you want to make sure that you have functional protein. Okay, this brings me to my last slide to just wrap everything up. The first protein structure was solved in 1959, then in 1980 the detergents were introduced. It took 5 years for the first membrane protein structure to be published. In 1996 we had the first ion channel, big success. And then in 2002 the nanodiscs, they were introduced. In the same year the first ABC transporter structure was in detergent solved. In 2007 we had the first protein in a nanodisc, and in 2009 then we had the copolymers introduced. Up to this year we had 200 unique membrane structures, so it took much long to solve like 200 different structures. In the year 2015 there was already 600, and in 2020 already 1,200 membrane proteins. So we are on a good way. It's now possible to make these structures. And with copolymers, it was possible to get the first sub-nanometer cryo-EM structure of a protein in 2018. And just in this year, just to put it in perspective, we have more than 85 copolymer-related publications just in this year. So this is really a field which is moving forward fast, and I can only advise you to join the team of copolymers. I'm convinced of them. They're really nice. There are some challenges, but you can be and you will be successful with this if you put in the time and the effort, and you have the advantages that you get the distinct membrane environment that stays conserved, talking of lipids and microdomains. Your protein complexes can be isolated and stabilized. Your copolymers, they will adapt to the hydrophobic core of your proteins. Big core still fits, small core still fits. With MSP can be a problem, and they are suitable for diagnostic assays. Yeah, and with this, I'm at the end of my presentation. I thank you very much for listening, and I'm now open for other questions. Okay, back to Lena.
Thank you, Philipp.
You're welcome.
For these exciting insights into synthetic copolymers. And thank you to the audience for all your questions. That's great. So as mentioned before, we will try to answer as many questions as possible now, but I think we will not be able to get them all. But let's get started with one question that we have 2 times regarding the polyhunter. And we have the question from Sylvie and Patrick, who are asking if the polyhunter also works for SMA 300 specifically and DIPMA.
We tried with all the copolymer classes, and for SMA, AASTY, and ultrasolute, you can get a depletion up to 99%, as I showed you. And for DIPMA, you're going to be around 60 to 70% depletion. So yes, it works. It works great.
Great, thank you. Ariel wants to know if we know how the polymers are interacting with the membrane or the protein, and which are the contact points.
That's a hard question. I don't think this is actually known in the community so far. So there have been several approaches to try to actually see where the contact points are, but how this is really working, sorry, I cannot answer this.
Okay, so then John has the question if the lipid composition of the expression host's membrane is important to consider when deciding which solubilization method to choose. You're on mute, Philipp.
Thank you, Lena. I was on mute. Yes, of course, it's super important that you try to keep the native environment as good as possible. So if you have a human membrane protein, it's best to also use a human cell line to express it. I can only advise you that how exactly for your specific protein of desire is influenced by the lipid environment. Of course, I cannot give you a distinct answer for this. We only know from publications, especially in the last 2 or 3 years, it really points out that lipid environment is crucial for function, for folding. There have been publications showing that also the opening of channels is only fully opened if it's directly interacting with the lipids. I think in this case it was the PC from the plasma membrane. It has to have the contact point so that the channel keeps open. So this was not resolvable in cryo-EM, just in DDM. So yeah, if you are able to make your workflow in this way that you try to really get into the original host, I can only advise you to do so. Yes.
Great, thank you. We got another question from Emma, who is asking if the synthetic copolymers are also available with, for example, an affinity tag, a biotin or fluorescent label, because, for example, the MSP-based nanodiscs are.
So far, no. So far, there are no alterations, but this is definitely much talked about in the SMALP community. Every conference we hear this demand, especially for biotin and fluorescence. I can tell you we are also taking a look into this, and I hope that in future we can support you not only with fluorescent and biotin labeled nano copolymers, but also with maybe affinity tagged copolymers. So far, they are not commercially available, as far as I know, anywhere in the market.
Okay, thank you. Sebastian is asking how stable these copolymer nanodiscs are. Is there any kind of shelf life? Are their temperatures stable? And there was another follow-up question from Sebastian. How would you start screening, and which buffer, which temperature?
That's a long question. A very good question. Thank you, Sebastian, for this. So this is actually impressive because copolymer formed nanodiscs are super stable. There have been experiments where they show that stably formed nanodiscs have a shelf life at room temperature of 60+ days. This is such a long time frame that nearly none of the membrane proteins I ever worked with or I ever know will survive 60 days at room temperature, but the nanodiscs, they will. So they are super stable regarding this. If you get them from us, they are lyophilized. You keep them in the freezer or in the fridge, you keep them dark, they're going to be stable. If you form them, obviously you will have your protein of desire in, then I can only advise you to work with them as fast as possible. If you can experiment with higher temperatures, so the second part of the question was, where would you start with the screening process? I personally would start at 4 degrees, and I would pick for a buffer medium salt, and maybe 20 to 50 millimolar HAPs, or 150 millimolar salt and HAPs, and check for the pH depending on the pI of your protein of desire. I would start with this, and I would start with 4 degrees, make my screening, and if I found some candidates which I like, I would then try to go up, because what we saw in our experiments is that sometimes higher temperatures up to 37 degrees can be beneficial for solubilization and also stabilization. So some membrane proteins, there have also been papers, are shown that they are more stable in 37 degrees than at 4 degrees. And another advantage of the copolymer form synthetic nanodisc with your membrane protein of desire inside are that they are more stable to freezing and thawing. So there have been experiments with a SMALP, I think 200, and a DDM-CHS, where after one freeze and thaw cycle, the DDM stabilized membrane protein was completely disintegrated, was not function anymore, and the SMALP conserved one was still working after 5 thaw and freeze cycles. I would still not advise you to just thaw and freeze it all the time. The best is to purify it and directly work with it, but they are very stable, just to say this.
Great, thank you. We have time for one, maybe 2 other questions, I think. We got the question from Sylvia. She would like to know how large a synthetic copolymer nanodisc is and how she can make polymer nanodisc also with lipids only. Is that possible?
Okay, yes, this is possible. So I saw a talk where they used just like lipids of choice, and they made the lipid micelles, they threw in the copolymer, and within a few seconds, then a stable nanodiscs were formed. Then, of course, they are empty, just filled with lipids. They will be in the range of 10 to 12 nanometers, most probably. But if they are surrounding your membrane protein of choice and you have a big complex, of course, they're going to be larger. And we can see that you really get a shift of membrane protein, as I also showed you in my talk, compared to detergents. You're going to get a shift to the bigger part with this. So depending on your complex, depending on the size of a membrane protein, depending on oligomerization states, you're going to get a shift. Empty nanodisc will be, the majority of them, going to be in the range of 10 to 12. And this is true for AASTIs, which are rough polymerized, so very, very narrow. And this is also true for the other classes. Even if you have, due to the chemical properties of the process, how they are synthesized, you're going to have larger ones and you're going to have smaller ones. We see, or it's proposed, that smaller ones and bigger ones can also work together. So the ring is not only formed by one polymer, it's formed by several polymers, and they can travel around. Also, the polymer rings, they can bump together and exchange lipids. So this is also super interesting, that you can see an exchange in lipids. If you put fluorescent lipids in one nanodisc and you have non-fluorescent lipids in the other, both are gonna glow at one point if you just wait long enough. So this is also interesting. It's a living system.
Okay, great. Thank you, Philipp. So I'm very sorry, but the time is, yeah, we're out of time now. We have a lot more questions, and we will be happy to answer all of them by email, especially the questions that are target specific. There are a few of them, yeah, that we got, so we will come back to you by email and try to answer your questions as good as we can. Yeah, we would like to thank you for joining today, and hope you enjoyed the webinar and you learned some things about synthetic copolymers. If you liked it, please visit us on YouTube, Instagram, or LinkedIn to learn more about Cube Biotech's products and services, and subscribe and like our channels. If you have any further questions afterwards, today, tomorrow, whenever, please feel free to drop us an email anytime.